MOFs are very interesting porous materials that they were discovered more or less about 20 years ago. These are made from a metal atom or metal cluster, which is connected with an organic molecule to another cluster, another organic molecule, and finally they self-assemble in a three-dimensional network. They have extremely high porosities. So some of these materials are able to reach 8,000 square meters per gram. Some years ago, people started looking at the possibility of encapsulating drugs inside the porosity of these materials and then to have a slow release. And what we are trying to do now is not only to work with the small drug molecules, but also to work with macromolecules, and in particular something like DNA or RNA macromolecules, so we can put them inside the porosity and we can protect them from enzymatic degradation.
In this work, we developed a MOF platform where you can encapsulate large amounts of small interferic ribonucleic acids, or siRNA, which is used in cancer therapy to knock down cancerous genes. And what's unique about the system that we developed is that the porosity of these MOFs is actually large enough to encapsulate an entire strand within it and really protect it from enzymatic degradation, because these molecules are incredibly unstable.
So the good thing is that it's highly specific. They don't have too much side effects compared to other very toxic chemo drugs. You can really tailor to specifically stop the propagation of one protein inside the cells and not the other one, so you only kill cancer cells, or you don't allow them to continue growing. We started looking for materials, MOFs, with biocompatible metals, and then we were looking also for MOFs with a porosity which is big enough to be able to accommodate this siRNA molecule. We use supercomputers to screen the porosities of all these structures and then find a material that has this porosity which is large enough for the RNA molecule we had. Because we knew the structure of the RNA molecule, is very easy to start inserting this molecule inside the porosity and trying to see if it fits or doesn't fit. One of the main questions as well we had is the siRNA molecule inside the porosity or is outside the porosity, just on the external surface of the nanoparticle? If it's on the external surface, the problem is that it can be degraded by enzymes, so we needed to put this inside. Using some super resolution microscopy techniques, we were able to analyze the localization of these macromolecules. We found that it was inside, we found that it was protected from enzymatic degradation, and then we were able to put them inside the cells.
We found that by using a proof of concept system that was based on fluorescence readouts, we were able to encapsulate siRNA within the MOF, deliver it to cells, and get approximately 27% knockdown. In addition, what we found was that by incorporating these extra endosomal release factors, we were able to let the contents within the MOF escape the endosome upon cellular uptake, and that was how we were able to see really consistent levels of gene knockdown.
Basically, it's a platform technology that we can use for almost any molecule and many diseases, not only for cancer. The good thing with metal organic frameworks is that we can really tune all the properties from all these materials. In the last 20 years, there have been about 80 to 90,000 different metal organic frameworks. So you can imagine how versatile these materials are. We can choose MOFs with different porosities, and they can be tailored to the properties of these molecules we want to encapsulate.
In this case, we've really shown proof of principle that you can use MOFs as a therapeutic delivery vehicle. And it really does set the stage for ongoing work where you can simply switch out the siRNA for a different sequence that codes for any cancerous coding gene.
If we focus on difficult diseases, these materials are going to have a big opportunity.